The overall goal of this procedure is to rapidly mix two solutions to start a biochemical reaction on the microsecond timescale. This is accomplished by first fabricating a microfluidic mixer. The second step is to bond a chip to a cover glass to seal the channels. Next, the chip is mounted to a manifold that contains the solution reservoirs. The final step is to image the mixed solution using confocal fluorescence microscopy. Ultimately, the biochemical reaction is captured on the microsecond timescale by converting distance along the exit channel to time.
The main advantage of this technique over existing methods like stop flow mixing is that mixing is not limited by turbulence and occurs in a few microseconds. This method can help answer key questions in the protein folding field, such as when does hydrophobic collapse occur? Demonstrating the procedure will be Ling, a postdoc from my laboratory.
To begin fabrication of microfluidic mixing chips, first clean 4-inch fused silica wafers with fresh Piranha Solution, as described in the written protocol accompanying this video. Next, use a low-pressure chemical vapor deposition furnace to apply a polysilicon coating approximately 1 micron thick per every intended 10 micron depth of the final microfluidic channels. Following further treatment of the wafers, as described in the text, spin coat the wafers with a 900 nanometer thick layer of AZ5214 photoresist. Then, soft bake at 90 degrees Celsius directly on a hot plate for 1 minute, or in an oven at 110 degrees Celsius for 30 minutes. After aligning a photomask, make a